So good morning, everyone, in Israel, in Brussels, and wherever you are joining us from to this webinar. Thank you very much for joining us. So my name is Jessica. Along with Smadar Hirsch, we are from ESERD, the Israel Directorate for European Research and Innovation, based at the Israel Innovation Authority. We're currently actually the national agency responsible for Israel's participation in the Horizon Europe program, of which the ERC grants are a part. Today, according to the demand and the request from the research community in Israel, we'll be focusing on the ERC proof of concept grants. So we have the pleasure to host this morning Laurence Colin, the call coordinator from the ERC proof of concept from the ERC Executive Agency. She will be presenting the application and evaluation process, meaning how to prepare and submit a competitive proposal, key evaluation criteria, and reviewer expectations, overview of the evaluation process, and common weaknesses and leading causes of proposal rejection. After that, we'll have two grantees from Israel, Professor Shulamit Levenberg from the Technion and Professor Igor Ulitsky from Weizmann Institute of Science, presenting from their experiences. From our perspective on a national level, I will say that the transition between the basic research and applied research is a natural transition in Israel, part of the Israeli DNA. The Israel Academy understands the importance and value of innovation. The TTO's units were established to maximize the value of innovation, and the ERC proof of concept are like the connecting link between the basic research and programs such as the EIC transition. And therefore, these grants are important and crucial for us. At this point of the 2026, we are between the first cutoff. PIs have received the evaluation a few days ago, and the results will be officially published on June 30. And we are currently heading towards the upcoming deadline in September 17. Technically speaking, this webinar will be partially recorded, meaning we will record the presentation, but not the question and answers sessions, according to the ERCEA's policy. The recording and representation will be sent to all the registrants to this webinar. This webinar was organized in collaboration with the Ministry of Innovation, Science and Technology in Israel. So we'll begin with the opening remarks by Professor Miri Yamini, Chief Scientist at the Ministry of Innovation, Science and Technology, and Professor at the Technion. So Miri, please.
Thank you so much, and thank you for having me here. I'm really excited to have this event going. I'll keep it short. As a professor at Technion and the holder of ERC Consolidator grant, I'm really excited to have this session, and I am heading currently the Israeli Fund for Applied Research at the Ministry of Innovation, Science and Technology. We truly believe in Israeli science and in the connection between basic and applied research. I urge you all to try and submit your applications and try to think how your amazing research can be applied and used for the need of society, for the need of economy in Israel and in the world. And I look forward hearing from Laurence about how to write and improve our grant proposals. So thank you very much.
Thank you very much, Miri. So Laurence, you can share your presentation.
Yes.
I will say that Laurence will be presenting about half an hour, 30 minutes, and then followed this part will be giving you the possibility to ask questions. So you can already post your questions in the Q&A box, and we'll be reading your questions after the presentation. So Laurence, we only have like this gray box on the top. If you
on the top, I know
yeah.
it's supposed to disappear in a few seconds, hopefully.
Okay.
Otherwise I can. Okay, I'm not sure I can myself remove it. So yes, it should disappear now.
Yes,
Is it okay?
it has.
Okay.
Yes, great.
Great.
Thank you very
Great.
much.
Thank you. So I don't see you. I don't see the question. So I will go through my presentation. If there is anything really burning, just
Okay,
talk,
no
because
problem.
otherwise I don't see any. So good morning, everybody. I'm Laurence Colin. I'm working at the ERC Executive Agency since 10 years now, but I was working in the starting grant team. And since 2 months now, I'm the new PoC grant call coordinator. So I'm very happy to be here for you to go through the proof of concept grants. So the presentation will be divided in 4 parts. First, how to apply, then a few data on the 2025 results, some information on dissemination and valorization, and some examples of PoC projects. So what are ERC proof of concept grant? It's a supplementary funding for ERC grantees. It's given as a lump sum of 150K for a duration of 18 months. And the aim of a proof of concept grant is to demonstrate that the ID funded by the original ERC grant has innovation potential and significant economic or societal benefits. As already explained, we have one continuous open call that opened on the 20th of January and 2 cutoff deadlines on the 17th of March. This is finished. And then the next deadline is 17th of September. The call budget this year is 60 million euro. So what are the objectives and the scope of PoC grant? The objective is to facilitate the exploration of the commercial and social innovation potential of ERC-funded research and to progress on the path from groundbreaking research towards innovation based on the idea from your main grant. We do not use the TRL in our grants because contrary to EIC, we do not ask you to be already around TRL 3 or 4 and to progress. We also accept proposals that are still at TRL 1, 2, and want to progress towards innovation. The evaluation is, as for main grant, based on the only criterion of excellence and divided into 3 main parts: the project and breakthrough innovation potential, the approach and methodology, and then the PI, strategic lead and project management. You will be, if you are granted, asked to provide a data management plan within 6 months from the project start, and the gender equality plan for your host institution is required as the signature of the grant agreement. What type of activities are funded under proof of concept grants? First, the activities should draw substantially on the ERC-funded research. It can cover activities aimed at exploring the path from groundbreaking research towards innovation, as I just said, including at very early stage of moving research idea towards an innovation or socially valuable proposition. It can include research activities to translate and promote research ideas into application, including testing, experimenting, demonstrating, validating the idea in the lab, as well as establishing viability, technical issues, and overall direction. It can also include clarification on IPR protection and the development of knowledge transfer strategies. It can involve stakeholders to help translate the innovation idea into innovation. There can be industry partners, societal, cultural organizations, policymakers, etc. And it can also include the assessment of potential end users of the proposed idea. First, the eligibility conditions. Eligible PIs have an ERC grant either running or that has ended after the 1st of January 2025. As I already said, the PoC ID has to draw on substantially on the ERC-funded research. But this is something that we are not assessing in the agency, contrary to the other eligibility condition. The fact that the ID draws on your main research is assessed by the experts. You can only apply once a year. So if you applied for the first cutoff, you are not eligible to apply for the second cutoff. There is a maximum of 3 PoC grants per ERC starting consolidator or advanced grant grantee. Those 3 PoC grants can run in parallel. And for the synergy PIs, the whole group of PIs in a synergy team can have 6 PoC grants. And if a synergy PI wants to apply, they have to have the consent of all the PIs in the synergy project. Your host institution can be different than the one for the main grant. It can also be the same. And you will need a host institution support letter at submission. And additional beneficiaries are allowed, similarly to ERC main grants. And also the principle of portability is valid for PoC grants. What is the evaluation procedure? So it's a single-step evaluation, contrary to main grant, with around 100 days between submission and notification of the results. The proposals are evaluated by 5 independent experts. They are not scientific experts, so the one that we have in our main panels, they are experts in technology transfer or social innovation. Of course, they have a scientific background covering all the ERC panels, but their main day-to-day work is in technology transfer and or social innovation. So we have 3 evaluation elements, and they do a remote evaluation, and they score each of those 3 evaluation elements. This year, we have 3 possible marks: very good, good, or fail for each of the 3 elements. Next year, it will change. And contrary to other grants, there is no panel meeting discussion. So the ranking is only based on the individual review. Panel members do not talk to each other about the proposal. Then, so we receive all the reviews. We rank the proposal based on the score. Of course, in between, we do a quality check, meaning that we read all reviews and we check that they address the evaluation criterion and that the score seems to be in agreement with the comment. If it's not the case, we go back to the experts and we ask them if they can confirm or maybe make changes. And then we have a ranked list that is based on the marks. And you have 2 possibilities. If you have a fail in one of the 3 criterion by a majority of the experts, your proposal will not be ranked. If you do not have a fail, then all proposals are ranked. And then we use the budget starting from the first proposal until we use the whole budget. And we create a reserve list in case of granting failure. So for those who maybe applied this year and are on the reserve list, we have quite a big reserve list because we have a bit of money that will be shared between the 2 deadlines at the end of the year. So what is a proof of concept proposal? It's a 10-page proposal, excluding abstract, references, and risk mitigation table, divided into 3 parts that are a mirror of what the experts will judge in their review. The first part is about the idea, the breakthrough innovation potential, maximum 3 pages. The second part is approach and methodology, maximum 6 pages. And the last part is about the PI, strategic lead and project management. Please just take into account that this last part on the PI, it's not similar to what you are used to write when you write an ERC main grant, because it's really on the strategic lead and project management skills of the PI and not on the academic, let's say, CV, even if it's also important to mention it, but in short. There is the online administrative submission form 8. It's the same as for main grants. And you can add supplementary documents. Host institution support letter is mandatory. And you can also add letters of support or letter of intent. And also, if needed, ethics or security document. sorry, you have here a link where you can find more information. We also have a YouTube video. So here are the evaluation questions. So what I would advise is when you write your proposal, you take those questions into account because this is what the evaluators, the experts will look at. For the first part of the proposal, so the first evaluation criterion, breakthrough innovation potential, experts will have the following question. Does the idea have the potential to drive innovation and business inventiveness or tackle societal challenges? Are the outcomes innovative or distinctive compared to existing solutions? Is the idea high risk with a potential high gain? If successful, will the outcome result in a breakthrough innovation? And is there a risk that some aspects are difficult to overcome? So this first criterion has a higher weight than the second criterion and then the third criterion. So this is really important criterion. And why are PIs failing this criterion? Either because this is not innovative, or what I mean is that the idea is still also in the lab. Even if I say that it can be at very early stages, it still needs to be different from what you have in your main ERC project. And you also have to be convincing that you are addressing a problem with a solution and that this solution is different or at least more competitive or with something new compared to existing solutions. And then regarding the risk, there is another place where we talk about risk, but here you have to show that even if it's high risk, the gain is important either for society, for economy. This depends on the idea, of course. Then the second part is about approach and methodology. So here again, you can see the questions. So I will not read, but you will receive the slides, but there are two parts. One is really on the methodology, how you will test or experiment your idea, what research you still have to do, or maybe you will only clarify IPR, you will start making a prototype. It's also important to include if you want to involve partners, industry or societal or cultural, policymakers, any other partners. And if you do, then make sure that you show that they really are involved using, for example, the possibility to add letters of intent or letter of support. And it's also very important to assess potential end users, because this criterion usually fails, it receives a fail mark by experts if there are no end users identified in the proposals. And the second part is about the timescale, the resources for the implementation. So you describe the team also. If you have names of people that you know already will work on the project, you can add the name and see and explain their expertise, for example. And for the timescale, very often PIs do include a Gantt chart. I see that this black thing is coming back, but yeah. And then the last part of the evaluation is the PI, the strategic leadership and project management. So this is the criterion that has the smallest impact on the score because it has the lower weight, but it's still important. And here you have to show that you have a clear vision on how to manage the project, the team, and to how you will take a strategic decision. You implement the proposal, and there is also a table on the risk management. And here, the main concern usually by experts are either that PIs are still on the academic part and that they do not have any view and or experience on the strategic and innovation part of the project, or that the PI is delegating all the work to someone else and has no Will leadership? This is what we see. So a few tips. Be convincing about your involvement in the project. It should be, the experts should be convinced that the PI will lead the project. You should know your competitor, describe the competitive landscape, what other solutions to the same problems are they already exist, and what is the advantage of your solution, what is also the novelty in your idea, who are the end users who will benefit from your innovation, and in case it's a commercially valuable innovation, you should describe the market. In case of a socially valuable proposition, then what will be the impact of your project ID when it's fully developed? In case of a database or application development, how you will maintain this beyond the project period. And in case there is additional funding or investment needed, how do you think you will pursue this in the future? Through which channels, maybe via industry, via other funding mechanisms that I will talk about later. So as I said, we have a list, a ranked list of proposals, and we use the budget that we have, but we also have very good proposals that are not funded because they are highly scored, but they are not sufficiently highly ranked. And all those proposals receive the Seal of Excellence. So a Seal of Excellence will be awarded to proposals that will receive an overall pass mark in all three of the evaluation elements set out in the work program, but cannot be funded due to budget constraints. And this Seal of Excellence, applicants will receive it in the FNT portal when they receive their results. They just have to agree to share information with other EU or national funding bodies. And what can you do with this Seal of Excellence? You will also, if you receive it, you will also receive guidance from RTD. RTD is our parent DG at the European Commission, as to where you can search for funding. And there are funding bodies that recognize and support the Seal of Excellence. You have a link there, you can go and see. And there are also some national, regional, or institutional ERC runners-up initiatives, for example, in Croatia, but I don't think it exists yet in Israel. So here I can show you that with their Seal of Excellence, ERC PoC holders can go to EIC transition. And you can see here in orange the percentage of proposals submitted from PoC to EIC, and in blue the success rate of those proposals. And as you can see, there is a quite high success rate. 2025 was slightly less good because first EIC was open to more types of applicants than before, and also the budget was importantly reduced. So I will quickly go through a few results so you have an idea of the success rate, for example, and the number of proposals we received. So in 2025, the budget was only 30 million. So, and the Scientific Council added 15 million of additional budget compared to now. This year, you know that from the work program, we already have 60 million. So it's double compared to the work program of last year. And you can see here the number of proposals received in total and per domain. It's the domain of the main grant. And as you can see, so we in total for the two deadline received 879 proposals. We could fund 300, and the success rate for the entire call is 34%. This year it will be normally similar. And because we received more, depending on how many proposals we receive at deadline 2, but at deadline 1, we received more proposals than last year. And you can here maybe see that social sciences and humanity apply less than physical sciences and engineering or life sciences, and their success rate is also lower than for PE and LS. If we look at the panel of the original main grant, you can see that there are panels that are more applying to PoC, and here it's the funded, but it mirrors the submission data. So in PE, the panels who have the more success rate and also who have the more proposals are P5, P7, and P8. In life sciences, it's the medical panels, LS4, LS7, and also LS2 with omics, let's say, and LS9, applied science. And in SH, it's mainly SH4. But this year we saw a few proposals from other SH panels. And from Synergy, we have less applicants usually. Last time we had only 12 proposals funded from Synergy PIs. Regarding the main grant, PIs who have a starting and a consolidator grant are more active in applying to PoC than PIs who have advanced or synergy grants, but the success rate is similar, as you can see here, except for synergy where it's slightly lower. For the other main grant, it's similar. And here it's the call type of the original main grant. So you can see that PIs usually, if I take, for example, consolidators, they do not apply or less apply in the first 1 to 2 years of their main grant, because probably they need to first have results on which they can build PoC proposals. And then from year 3, it's more or less the same until the end of the eligibility window. If we go to the funded proposals for PoC2025, you can see here per country the number of funded proposals, and you can see that Israel is here. So after those countries, but quite well positioned before all those countries. And here it's the success rate by host institution country. And this is in red, you have the average success rate, and you can see that Israel is above average. Here it's all funded PoC projects from 2011 to 2024. And you can see Israel here with around 180 PoC grants. If you are interested to see what type of research is funded in PoC, there was a study for all PoC grants from 2014 to 2023, and you have the link here. You can go and have an idea. I will now say a few words about dissemination and valorization of ERC results. A few key concepts. So we aim at guiding and training applicants and beneficiaries by offering dissemination and exploitation capacity building activities, and motivate beneficiaries to scale up their results, try to synergize with other EU programs, for example, EIC transition, and support beneficiaries by offering some services. What type of services? The main tools and services to beneficiary. This is not only to PoC beneficiary, it's also to main grants beneficiary. We have the Horizon dashboard. It's a tool with data and statistics to explore the participation to the framework program by topic, country, region, etc. We have Cordis. It's an online platform with all funded projects and their results. We have the booster. I will get back to it. It's a free personalized service to support dissemination or go to the market effort. The Horizon Result Platform. It's a platform for promotion and matchmaking of research results with different, the relevant audiences, investors, researchers, policymakers, and so on. And we also have the European IP Help Desk that provides support, building capacity to manage IP assets by broad range of material, help for support on-site and online training, to which all grantees have access and for free. So the Horizon Results Booster, it's for all EU-funded research projects from FP7 onwards, and it's free of charge and tailored to your needs. The services include dissemination support to assess and improve dissemination plan and methods, and to identify appropriate audiences and channels, and the go-to-the-market support to increase the maturity of the results, to develop business and exploitation plans, and to identify exploitation opportunity of your results. So I think the link is there, so don't hesitate to go and check. And then I have a few examples of PoC projects. I took one in LS, one in P, and two in SH. Why two in SH? Because as I explained, SH is struggling to have high success rate, but also they are applying less. So we want to also tell SH grantees that they are welcome to apply. So the first project I wanted to show is Icarus, or Icarus, from Dr. Amit. So it's a PoC 2025 project that is based on an LS2 project. And I will give you just a few info. So autoimmune diseases such as rheumatoid arthritis and multiple sclerosis frequently progress despite treatment. And what they propose is an inducible CAR T-cell platform, which will be a game changer for patients in terms of quality of life, relapses, reduced treatment burden, and lower infection risk. So in the project, they are doing the next step of the research, meaning that they are developing this platform and they are testing it, and they are exploring a potential way to patent, etc. Vrai Cure, it's a PoC 2025 project based on a PE8 project from Dr. Eli Hakem. And this project aims to develop a clinical prototype ready for AI-driven clinical decision support for patients with aneurysm. So they had developed in their main grant an AI tool to have a real-time simulation of hemodynamics and virtual visualization of the aneurysm. And they know in the PoC propose to go to hospitals with this tool to help doctors take a decision when they have patients with aneurysm. Modern Argonauts, this is a PoC 2023 project based on an SH5 project from Dr. Marcignac. And she's showing that the questions that were posed by the ancients are still relevant, that the shared language of mythology creates a sense of community and hope for the future. So she developed a game that can be where you are in modern Argonaut world. You embark on an expedition, and the students can learn about ancient sources. They are doing games in different contexts, and they are selecting teams from other culture. And finally, Dream Code. It's a 2025 project based on an SH4 ERC project from Dr. Rinaldi. So this is a project on a non-invasive method to decode dreams using EEG recordings during sleep. And here they are using individualized decoding model and computational model, capturing the structure of memory. So this was my presentation, and I'm happy to take questions, but I will stop sharing so I can see you, hopefully. Yes.
So thank you very much, Laurence, for this comprehensive presentation. So we'll move to our next speaker, Professor Shulamit Levenberg from the Technion. She will be sharing from her experience. So Shulamit, please.
Okay, so I wasn't sure was it would be best to present something or not, but maybe I give a little bit of a background and then I will present so it will be more clear what the project, the ERC project and the POC that emerged from it. I had in my first ERC, I had the POC, and now in my second ERC, I received 2 POCs. And submitted another one. So So the idea was that the ERC was a larger project, and then throughout the project we developed things that we thought can be commercialized. And it are not maybe the main idea of the ERC, but they are development that could be commercialized, and they were coming out of the ERC, and that's why we submitted them as POC. So maybe I will share the presentation just to give you the idea of the concept of the ERCs and then of the POCs. One second. Okay, so do you see the presentation? Is it okay?
Yes, we do. Great.
Okay, so the idea is to bioprint 3D tissues with blood vessels, and the idea is to have tissue flaps, meaning that we can come with a large vessel, engineer the large vessel, and also the smaller capillaries. And the concept was that we can get connection between the large vessel and the small capillaries, and we can create the larger tissue. So this is in my first ERC, we were focusing on the small capillary network and how we can introduce it into different tissues. But then in the second ERC, the concept was different, how to really make large flaps. And the focus was on bioprinting and how we can combine large vessels to the small capillaries and create a multi-layered tissue. So the concept was to create this large vessel, and then it will be connected to the small vessels, as I described. I will not go into the details. And for this, we really came into the bioprinting and printing. As you can see here, this was published in Advanced Materials. This is the result of the ERCs, of having the large vessel bioprinted and connected. We demonstrated here in one strategy. And then we moved, okay, we showed the concept, but we want to do the multi-layered tissue as we describe in the ERC, to have skeletal muscle, to have fat, to have bone, to have all this tissue connecting with the large vessels and everywhere connecting to the small capillary structures. And you can see some other work in this concept that we've showed it with adipose tissue, bioprinting, and now more branching, vasculature, still the combination of the large vessels and the small capillary and the connectivity between them, and being able to do it within adipose tissue, functional adipose tissue. This is another example of creating into skeletal muscle and using it to repair volumetric muscle loss, large muscle injury, and again the large vessel and the smaller capillaries. So just to give you the idea, and now the multi-layer that we wanted to do. So here is adipose tissue and muscle tissue together, multi-layer with a flap, with the idea of large vessel connecting to artery and vein, and also getting into the implantation. This is small animal suturing the artery and vein. All this is fully bioprinted, engineered, muscle, fat, and all this, and also moving to larger animals, the sheep, for facial reconstruction. And it involved also building the bioreactors and printing the tissue, building it, and analysis of the tissue, and also the implantation. But during this, I will not show this movie, the concept. So during this project, we really went deep into the bioprinting. And when we went into the bioprinting, we realized that there are tools that are missing, and we had to develop some of the tool to improve our bioprinting techniques. And so here you can see an example of printing into a printing bath, and this is important because our POC was on the first POC was on developing a new printing bath. Okay, so here is the concept of a printing bath, that we have a material, if we're printing a soft polymer, so we can print, we cannot print in air because the polymer is soft. So we have to print in a printing bath that allows us to print the different layers and only at the end polymerize them. Because if we print layer by layer, we have to wait for a long time for the polymerization, and the polymer is too soft. So the printing bath was developed in the field, not by us, but this is by Adam Feinberg, and he used gelatin-based particles, and that they are melting in 37. And what we realize that we now have to, if we print in this printing bath, and then it dissolves in 37 degrees, then we are left with the soft hydrogel bioprinting and everything collapses. And this is actually something that everyone in the field saw, but didn't have a solution. And so we can print very nice structures and then everything collapses. And during our printing of the tissue, we printed the muscle, we printed the fat tissue, we realized that we have to find a way to prevent this collapse because the shrinkage is not good. As you can see here, especially if we focusing on printing blood vessels, we need the lumen. If everything collapses, we lose the lumen. So we decided to develop a new printing bath. So the printing bath is made of kappa carginan particles. It's not melting in 37. And we can keep the construct inside it after printing. And this is where we call it print and grow. And this was a development that we submitted the POC on it, because it was, as you see, it was merged out of the ERC, but it's something that can be relatively fastly commercialized, and this is why we wanted to bring it closer to commercialization with the submission of the POC. And so this is the concept, that we have a new printing bath. We printed it with different polymers, and we show that we can keep the structure. As you can see here, we can keep the structure. If we keep the tissue in the printing bath after printing for, let's say, 1 day, 2 days, we can keep the structure, unlike if we remove the printing bath immediately after printing, as most of the researchers did till we develop it, then we see collapse. If we keep it in the printing bath, then we keep the tissue. And this was very important because you saw the structure that we wanted to print with the blood vessels, large tissue construct. We need to, and we don't want it to shrink. So this was the first POC, and indeed for the POC, we showed that we can large scale the production. We contact different companies that showed interest in this printing bath to sell it, printing companies of printers that also sell different bio ink and different printing baths and different materials. So we contact these companies and got a letter of interest that they want to explore this. They want to test it with their printers, and this is one POC on the printing bath. And the other POC is when we started to work on bone bioprinting, we realized that we need a stiffer bio ink. And all the hydrogel that you print with for biological application are very soft because the cells need this soft material. And we cannot print next to it a stiffer polymer because it either needs a higher temperature or organic solvent. And you cannot print like one head and one next to the other, the stiffer bio ink and the bio ink with the cells. And this is why on the way, because we wanted to print bone, we had to develop a new bio ink. And this is the bio ink that we developed, that is made on particles that can sinter together, because the way we make the particle, will not get, of course, into the details, but you can print particles because they are kind of fluidic, like when they are small particles. But after printing, and in 37, and also using the printing bath, we can sinter them, and they become a stiffer material that we can now use 2 different heads and print one next to the other, the bio ink with the cells and the stiffer bio ink that provides the right stiffness that we need for bone tissue engineering. And this is some of the particles. This was the concept of the second POC, was how we can make now the particles even better. We have particles, the particles are porous, and this also brings porosity to the structure, so medium can go everywhere. And we thought that the bioink by itself, because it's it's featured, and we submitted a patent. We also submitted a patent on the printing bath, I have to say. So we submitted a patent on it, and we also thought that we can add to it growth factors because it's porous. So these are porous microparticles. We can add growth factor into it, and we can now have a platform of particles, bioink based on particles that can provide stiffness and also can provide bioactivity. And this was the product that we wanted to develop, and this is what we wrote the POC on, the PLGA hydroxyapatite particles that are bioactive, that we can also incorporate different growth factors that can help to differentiate the bone, and growth factor that can help to differentiate the endothelial cells. And we have preliminary results on the particle that we can print them with cells, and we already had some data showing that we can implant this the construct with a new bioink. So this was also something that we contact companies that showed interest in this bioink, that they can sell it. So either we can commercialize a product or have a startup company for it. We left the two options in the POC to explore, but we already had companies that were interested also in licensing it. So I can stop share here. And though I don't know if I talk too long, if we need to leave time for questions, or if you want to elaborate more on specific aspect of these two POCs.
Wow, you did great. Thank you very much, Shulamit, for this very impressive and inspiring and clear presentation. So thank you very much. And maybe if people would like to address you privately, of course, they're very invited to. I don't see any questions in the Q&A. So thank you, Shulamit. We'll now move to Professor Igor Ulitsky from Weizmann Institute of Science. Igor, would you like me to share the screens, or do you want you to do it? You prefer?
I'll share the screen. And
Okay.
so thank you for the invitation. Actually, I learned a lot already. So I think the best advice is to tune into these webinars, which I didn't do. And I'll share a slightly, I think, different perspective, because my lab is, I think, in its nature a bit less translational than Shulamit's. But nevertheless, we gave a POC grant, and we were successful in one and not less successful in others. So I think I can also share some of the perspective on sort of what I learned in the process. So just to briefly introduce kind of the field that we're working on. So we're trying to address genetic diseases where people have a mutation, and in a subset of these genetic diseases, people have a mutation on one of the two chromosomes. And while for many genes it's okay to have a mutation in one of the two copies, for some genes it's not okay, and these are haploinsufficient disorders. And in these, typically, basically not enough protein is being produced, but some protein is being made. So what is desired is to make sure that we somehow make more protein, more than 50%, and go from 50% to 100%. And we're using a technology which is called antisense oligonucleotides, which is a way to go in and target specific RNA molecules, and this way to change their activity or their processing and splicing and so on. And in some cases, these can change protein production. And again, in our case, we're interested in going from 50% protein production to 100%. And importantly, this technology already has some FDA-approved drugs. There are some successes. There are some companies that are interested in pushing this forward. So kind of the relevant stakeholders are in general out there. And if we look at sort of the field broadly, then this is successful in some types of modulation of genes, but there is a need for new ideas on how to take this particular methodology of these antisense oligonucleotides and come in and use them for things that they cannot do successfully until now, right? In our case, in general, with antisense oligonucleotides, it is easier to turn off gene expression, and it's more difficult to turn gene expression on. And this is kind of the concept of what we're doing in general in the lab on this more translational front is try to figure out how we can do that. And our main discovery in this direction in recent years was that we can use these antisense oligonucleotides to basically form a connection between separate RNA molecules. So we're looking at a gene that doesn't produce a protein that we discovered, it's called chaser, and we're looking at a gene that does produce a protein and in which there is a mutation in a genetic disease, which leads to epilepsy and autism. And we've shown over the years we can use these antisense oligonucleotides to target these gene that doesn't code for protein, and this way to increase the activity of the gene that does cause for protein. So we published it, some of it is in bioRxiv. We have patents on this approach, and the first patents were licensed by a tech company, and they're trying to now sort of push it forward. So this is kind of the overall idea. And obviously, so we have this kind of first proof of concept on this particular pair of genes. And now we're trying to apply it to other gene pairs, which can also benefit from a similar approach where we can target one gene and this way increase protein production from another gene that is found nearby. This is kind of the overall idea. And in the Fusion ASO proof of concept, which was successfully funded, we're applying this to Wiedemann-Steiner syndrome, which is another haploinsufficient disorder where there is interest of increasing gene expression from a gene called KMT2A, and we are interested in doing so by targeting a gene called ATP5MG. So in this case, when we applied for this funding, we haven't had any results yet on this pair of genes, and we had results on chaser and CHA2. So my own history with ERC. So we had an ERC starting grant, which was called Link Safari, which was more sort of basic research into the evolution of these non-coding RNAs. And this is sort of how we got started in working on chaser, which is the RNA that we're targeting. And then we had just finished an ERC consolidator grant, which basically was more of a diving into the mechanisms for which long non-coding RNAs are functioning, including this chaser and CHA2 as a kind of circuit of particular interest. And we tried to apply for proof of concept 4 times and got 1. So in terms of success rate, actually, we're a bit below the average. But I think it can be a useful perspective on that, where there were 2 applications which were on this initial circuit of chaser and CHA2. They were kind of earlier on. We didn't have IP yet, and they were more about sort of trying to understand better how this circuit can be tuned to increase gene expression. And they were not successful in part because of that. We didn't have IP yet. It was more of sort of funding for more research which was required to make it applicable. And then we had a successful application in 2023 where we could already show that we have this working, we have IP, there is a company that's licensing the patent on this first idea, and now we're extending it to another. So this was more successful. And more recently, there was another unsuccessful application kind of in a similar spirit where we had another project where something was working and licensed, and we wanted to translate it further. It got good reviews, but not excellent reviews. We got this seal of excellence, actually, but it wasn't funded, so we'll probably try again this year. So this is a typo. One successful, this is not that. So in terms of how we were writing these, so actually twice we used a company called Accelerate that the Weizmann Institute is working with. I think others are working with this as well. This is a company that helped us formulate the grant. I think it's important. You can also see it from the discussion. I think the biggest difference for me as a researcher in writing POC versus other things is that the text is really different because the reviewers are really different, right? We're really used to writing academic grants where the reviewers are people working in an academic environment, which are evaluating the research. And I can also say personally, I really kind of, it's easier and more enjoyable for me to write these kind of grants. ERC PoCs, as you heard, reviewed by obviously people with academic background, but that are not other group leaders or other PIs. And so really the writing needs to be tailored for that. And I think for us as academics, this is a highly non-trivial exercise. I can also say personally, I enjoy far less writing in this spirit. So it was really helpful work with this consulting company that assisted with it. Again, in the Weizmann, there is a scheme where the institute is supporting some of the associated costs. In principle, always, I think it's better to start working with them earlier on. In my case, it was quite late in the game, but they basically discussed the idea with me. They prepared some baseline, and this was a part of sort of the overall envelope of a grant. Again, particularly the overall kind of envelope addressing all of the things that we are not used to write in grants. And again, if you haven't wrote a POC yet, it's really a very different experience because we're not used to that. And then twice I applied kind of independently, building on the previous kind of on building on some of the envelope that was prepared elsewhere. So I think one thing that is useful, and I sort of copy pasted some of the criticisms that the ones that were not funded received, and you can see another sort of key thing I think which is very different is that whereas in regular grants that I'm reviewing and that I'm submitting, there is always some guidelines that the reviewers get about sort of what's important, and sometimes they work with these guidelines. And if you submit an ISF grant, sometimes you would get kind of a response that is structured according to what the agency is asking the reviewers. But in many cases, that's not the case, right? When I'm reviewing a grant, typically I don't actually read what the agency says. I read the grant and I write a free text about kind of my impression of the grant. In my impression from the review of the POC is much more structured, and the reviewers are working with the specific items that were mentioned in Laurence's talk earlier. So it's really important that what you're writing is actually addressing these individual points. And again, the consulting company, again, you can work obviously with anyone, they can help with that because obviously they're more experienced. But as you can see from some of these reviews, and again, I'm not going to kind of read them because these are examples. They often address specific drawbacks or specific things that were missing from my proposal, even though in some cases they were not missing. And these are things that we're not used to necessarily writing. So, for example, there is limited time dedicated to marketing research and commercialization activities in the plan. I think in this case, there were some things in the proposal, but again, these are not things that I'm used to writing in the grants. They comment on decision strategy that is not really clear. Again, not something we're used to writing in grants, and so on. So, for example, and again, the focus really needs to be on the transition to marketing and translation and so on. So, in the second time I tried, which is really, I think, the worst reviews I got, there is a major scientific focus on the explanation rather than on translation and commercialization, right? So they kind of caught that I'm really interested in the basic research, at least at that point, and it wasn't clear enough how this is commercial. So I think my impression again from this experience is compared to the grants that we're used to writing in academic setting, it's really important that there is a detailed execution plan that fits the time frame of the grant. It's important and that sort of there is track record of the tech transfer office, how good they are at it. If the PI has a track record in translational research and so on, this is also something that they're looking at. As Laurence mentioned, it really helps to show that the PI is really kind of on board with this commercialization or societal impact strategy. Again, in my limited experience, it really helped that we had already some IP and some licensing, not necessarily on that particular project. So Fusion ASO, we're proposing something on a pair of genes we haven't worked at before, that we haven't had IP on before, but we could say this is a direct follow-on on a project where we do have IP and do have licensing. Kind of important to emphasize, on a project we already have licensing, you typically cannot really apply for a POC, right? Because that project is kind of already progressed beyond the point where where sort of POC is relevant. But it really helps if you can show we already did this successfully, we already got stakeholders engaged on something, and now we're going to do something else, which is related, because then it's easy to convince them that you're really in the game. Again, these detailed plans for decision-making, for engagement, really having an alignment, I think, with what the criteria for evaluation are, is more important here, from my experience, than in a typical academic grant, because the reviewers are more likely to work vis-a-vis with the criteria that they're expected to evaluate based on. And as already mentioned, sort of a defined market. So the stakeholders, in this case, what the companies that are relevant. I don't think I included any support letters, but obviously, kind of hearing Laurence, I think it's that can be important as well. So a clear audience that will eventually, after the POC, be the one pick up the tab for continuing the project. I think is really important. That's what I had to say.
Thank you very much, Igor, for this inspiring and to the point presentation. I'm actually positive that your inputs really set the example for future applicants and hopefully grantees. So thank you very much. So for dessert of this webinar, we'll move to Smadar Hirsch, the ERC manager, director of the ERC grants. She will be talking about the closing remarks and key takeaways ahead of the upcoming proof of concept deadline in September. And I will just say that if you still have more questions, you can always, post the question here in the Q&A. And also, I just post in the chat the ERC functional box of the team, so we can always post your questions in, send an email. This is the email. Okay, so Smadar, please.
Thank you very much, Jess, and thank you, Laurence and Shulamit and Igor, and of course Miri, for a fabulous info session today. The webinar is very good. I would like first maybe to ask Laurence, if possible. You were asked a question which you wrote, but I think it would be interesting for the others to know as well. So if you would be willing just to share, we will have to stop recording. Okay, thank you. I would like just to say that when you look at the Horizon Europe, you can see that actually the ERC starts from the basic science, and there are programs that are all the way to the SME, the small and medium enterprises. The first step is a proof of concept. And then, of course, there are pathway and then the EAC accelerator and so on. So this is an excellent opportunity to try and see if your fabulous ERC, starting, consolidator, advanced, or synergy, has something which could be commercialized, but not even look at the commercialized. Think about the way you can contribute the society, find solutions which are so essential to our society, and then think of taking advantage of this grant moving forward to a more specific, towards initiating and having an SME. That's the first thing. Second, I hope you understood that even though it is a different grant, you still need to invest time and consider various aspects. This time, not only the scientific aspect, but look at the target audience, who will benefit from it, and what can be gained, what is the impact of such a project on the society. And then consider all these aspects just before submitting a proposal. So this time, the ERC asks you to look at different angles of your research. And I think this is the most important aspect of this grant. I hope many of you will take advantage of this project. As you could see, Israel is doing rather well in this type of grant. And you know, it is not new to us that from the academy, people are familiar, I would say, with this. It's part of, I would say, part of our DNA to move from basic research towards implementation of solutions and innovative solutions. So please take advantage of that. And of course, the most important thing is you've heard Laurence, you have the functioning mailbox, you have us, you heard the very inspiring success stories. Take them all and don't hesitate to contact us for further assistance, advice, whatever. That's it for my side. I wish you great success, and we will be very happy if you have more questions. This is the time to post the last questions. Let's see.
Thank you very much.
Yes, it looks like at this stage, all your questions were asked and answered. So good luck and have a fabulous day. Thank you for